Tay-Sachs and GM1 are very similar diseases. We consider them to be like sister diseases because they're extremely similar. The clinical signs are almost identical in kids. Kids begin to have clinical signs at 6 to 8 months of age. They start losing developmental milestones. Parents usually have no idea that they could be carriers for these diseases, and so it takes another 8 to 10 months to get a diagnosis, and by that time the kids have progressed quite a bit from their original disease onset. The basic gene therapy is the same for both diseases, but because of a little bit of a strange biochemistry with Tay-Sachs disease, we have to use 2 different gene therapy vectors. We call the treatment agent itself a vector. For that reason, we really need to inject the brain directly, because if we injected the bloodstream, then those 2 vectors would get diluted out so extensively that any given cell would not be treated by both vectors. It's been far beyond anything that I ever expected. I thought we might be able to extend the cat's lifespan by 50% and maybe get rid of some of the most severe signs of the disease in the cats, like they have a very pronounced whole body tremor. And I thought maybe we could do something about that. But we've extended the cat's lifespan 4 to 5 times, and most of their symptoms are completely gone. Their quality of life is dramatically improved, and there's no question that if the results in the animals translate into humans, even to some degree, it will be a tremendous advance forward.
Auburn University is the only place in the world where you are able to study the Tay-Sachs sheep. My research focuses on optimizing the gene therapy protocols for the Tay-Sachs sheep specifically, and then we go back and look at the Sandhoff cats and the Sandhoff mice and see what the differences are therein. I've spent a lot of time doing a lot of MRI work, CSF analysis, blood analysis, trying to find objective measures of disease in the cats that also will correlate over to human patients. And so in collaboration with Dr. Tift at the NIH, we've established several different kinds of markers that are MRI-based biomarkers, CSF-based biomarkers that are the same in the cats as in the human patients, the untreated GM1 cats. And then we went back and looked at those biomarkers in the gene therapy treated cats to try and get an idea of how they would change after treatment and if the changes reflect the disease status in the animals. And we just published a paper on that. Actually correlates really nicely, surprisingly. So we have what we need to move forward to clinical trials as far as objective measures go. We have what we believe we need to do that.
It's really been a journey that's gone on for 4 decades. When the animals were first discovered, the diseases were first discovered in the animals. Obviously, what you wanted to do was find a treatment for the animals and ultimately one that would be applied to humans. All we need to do now is translate that into a human clinical trial and see if it works as well in kids as it does in the animals. The only thing that we need to do that is a little more funding to get the last couple of steps done and get the application submitted to the FDA for approval of that clinical trial, and then we're hopefully, and I believe so, off to the races.
I just want to get this to clinical trials. I'm not really looking much past that at the moment. I mean, we are very focused to take what we have, which is really the best results anybody has ever had in a large animal model ever. I mean, nobody has really reported the results that we've reported in a neurologic disease in large animals. And being able to take this efficacy over into human patients and actually make a difference in these kids' lives is unbelievable. I mean, we go to the family meetings every year. I know a lot of family members. We have lost a lot of children that we've known over the years. It weighs on our hearts heavily, and the only thing we want is to give this therapy a try and see how it works for kids. That's it. That's all we want.